Hey everyone, welcome back. In today's video, we're going to talk about VCF files. Let's get started. So what is a VCF file? It stands for Variant Call Format, and it's a file format for storing sequence variation information about single base substitutions, insertions, and deletions. And these files typically end with a .vcf file extension. So where do VCF files come from? They're produced by taking the reads in our FASTQ files and aligning them to a FASTA formatted reference genome, and then analyzing those alignments with specialized tools. And these tools look for differences between the reads and the reference to determine what kinds of variation exist. Things like single base substitutions, insertions, or deletions. And all of this information is stored in the VCF file. And the process of identifying these variants is called variant calling. And there are numerous tools you can use for variant calling. And I'll make sure to include links to popular variant callers in the video description. Now let's talk about the VCF format and how variants are represented and stored. The VCF format is a tab-delimited text file with 8 mandatory fields, which capture essential information about each variant. The VCF format also includes a mandatory header section describing the version of the file format used. There are also optional fields that provide extra details about each variant, such as genotype, the amount of reads supporting the variant, or information about its clinical and functional impact. And just like in our video on the SAM file format, we'll focus on the mandatory fields of the VCF file. The official VCF format specification includes a lot of information about each field, as shown here, but it's not that complicated. Let's take a look at an example. Here's what the first few lines of a VCF file might look like. The first line specifies the VCF version, here version 4.2. The second line lists each of the 8 mandatory fields, and each subsequent line corresponds to one or more variants observed in a specific location in the reference genome. Now let's go through the mandatory fields one at a time. Chrome stands for chromosome. This is the name of the reference sequence where the variant is located. In this example, chromosome 20. But this could be any genomic feature from any organism. A gene, a plasmid from a bacteria, or even the name of a virus. POS stands for position. This is the exact position in the reference sequence where the variant is located. ID stands for identifier. This is a label for the variant, often taken from databases like dbSNP, which provide additional information about the variant, such as allele frequency, clinical significance, and name of the gene where the variant was identified. And if the variant isn't known or cataloged in a database like dbSNP, you'll see a period. Ref stands for reference. This is the reference base. And for this specific variant at position 14,370 in chromosome 20, the reference genome has a guanine, or a G. Alt stands for alternative. This is the alternative base. And again, for this variant at position 14,370, the reference gene has a guanine, but we observed an adenine, an A. This would represent a single base substitution. Qual stands for quality. This is the quality score of the variant, with higher values indicating more confidence that the variant is real and not a sequencing error or due to some other kind of technical artifact. Filter stands for filter status. This field tells you whether or not the variant passed quality filters. This could be user-defined, specified by you, the bioinformatician, or set by the variant calling software. For instance, one variant might fail if its quality is below a threshold of 10, as shown here, while others pass because their quality is above 10. Info stands for additional information. This field provides extra information about each variant and can include things like allele frequency, read depth, predicted impact on gene expression, or even a description of its clinical significance. Info entries are separated by semicolons and can get quite long. And if you're interested in learning more information about each subfield, or taking a deeper dive into the VCF format, please make sure to take a look at the official format specifications. There's a lot of great information in there, and I'll make sure to provide a link in the description. So what are VCF files used for? They're used to store genetic variation information, and we can use this information to study the impact of those variations on things like gene expression, to understand the prevalence of variants across different groups or populations, and to identify variants associated with human health outcomes, among many, many other use cases. Now, if you're generating VCF files for your own study, you'll want to know if those variants have been seen before and what their functional or clinical impact might be. And there are reference files in VCF format for exactly this purpose. If you're working with vertebrate or non-vertebrate genomes, you can find reference VCF files in the Ensemble and Ensemble Genomes databases. If you're working with human data, you can find high-quality reference VCF files and variant annotations in the Single Nucleotide Polymorphism database, also called dbSNP. And for human variants with potential clinical significance, you can annotate your variants using the reference VCF files in the ClinVar database. And before we wrap up, one of the things that I think is important to know is that VCF files aren't just used for studying genetic variation in humans. They're also widely used in infectious disease research. For example, researchers in the UK and Australia recently developed an experimental and quantitative method to identify sequence variants in bacteria that confer resistance to antibiotics. And these sequence variants are stored in VCF format and then used for further downstream analysis. And if you'd like to learn more about this research, I'll post a link in the video description. That's it for this video. In the next one, we'll talk about GFF and GTF files. I'll see you there.